Have you ever wondered how we actually measure the protein content in grains or food samples? It's not just for nutrition labels, it's crucial for food quality, animal feed, and even international trade. Now, you might be thinking, can't we just look at the ingredient list? But it's not that simple. Proteins aren't visible to the naked eye, and their content varies with every crop batch. In food science, we use a chemical approach that targets nitrogen since proteins are rich in nitrogen atoms. This is where the famous Kjeldahl method steps in. Think of it like using a detective's toolkit. We're tracking down nitrogen, converting it into a measurable form, and then calculating how much protein must be present. By the end of this episode, you'll see how a series of clever reactions, some bubbling chemistry, and a dash of math unveil the hidden protein inside every grain. Let's dive into the first stage, digestion. Imagine dropping your grain sample into a cauldron of concentrated sulfuric acid. Sounds dramatic, right? That's essentially what we do. This step breaks down complex organic matter so we can target just the nitrogen. Chemically, sulfuric acid acts like a powerful solvent, oxidizing everything but the nitrogen. That nitrogen is transformed into ammonia gas you probably remember from household cleaners. But here's the twist. The reaction needs a little help. This is where catalysts, or helpers, like copper sulfate or selenium come in. They speed things up, making sure all organic matter is fully digested. Picture it this way. If the sample is a locked box, sulfuric acid is the key, and catalysts are the oil that makes the lock turn smoothly. Once digestion's done, all the nitrogen in your sample is now ready for the next stage, liberated as ammonium ions waiting to be measured. On to stage 2, distillation. Here's where things get interesting. We need to separate the ammonia from the digestion soup. How? By adding sodium hydroxide, or NaOH strong base to neutralize leftover acid and convert ammonium ions into ammonia gas. Now, you might ask, why make ammonia a gas? Because gases are easier to collect and measure. It's like letting steam escape from a kettle and catching it in another pot. This escaping ammonia gas is captured in a precise amount of hydrochloric acid, a process known as absorption. The technical term here is quantitative transfer, which just means we're collecting as much ammonia as possible. And to make sure we don't miss the endpoint, we use a color indicator. Think of it like adding dye to see when your tea is strong enough. By the end of distillation, all the nitrogen from the original protein is now locked in a new solution, ready for final measurement. The final act, titration. This is where chemistry meets math. We now have our captured ammonia and acid, and we need to figure out exactly how much is there. We do this by adding a known concentration of sulfuric acid until a precise color change, usually purple, signals the endpoint. You might be asking, how does color tell us about protein? Well, the color change marks the chemical equivalence point, meaning all the ammonia has been neutralized. This is a classic acid-base titration. Now comes the formula. Ready? Protein percentage equals your titration reading times the normality of acid times 0.014 times 6.25 divided by your sample weight, then multiply by 100. That 6.25 is a protein conversion factor, an industry average for how much nitrogen is in typical proteins. It sounds complex, but picture it like weighing a cake recipe. You need the right ratios to get the perfect result. So, by the end, we've turned a mystery sample into a clear, actionable number, percentage of protein, ready for labeling, quality control, or research.